Introduction, and thanks everyone for taking the time to join us today. So as Gabrielle mentioned, today I hope to review some of the small animal preclinical imaging modalities that we represent here at Scintica. The focus will be on the modalities and less on the products themselves. And then we'll spend a bit of time talking about multimodal imaging, the importance of it, and review some papers that use multimodal imaging techniques. So what is preclinical research? So just trying to get everyone here on the same level. So with preclinical research, we're talking about both translational research as well as basic research. And the main goal typically is to uncover disease mechanisms, study the effect of a target therapeutic compound on that disease. And most often the goal is to improve both clinical diagnosis as well as treatment of those diseases. So when we're talking about preclinical imaging, what are we talking about? Most of the time, the imaging modalities that we'll talk about have been adapted from the clinic. So most people, when they go to the hospital or go to see a doctor, they may have to get an MRI, a PET, CT, or an ultrasound. Well, it's important that we be able to use those imaging modalities in preclinical research so that we can translate the findings into the clinic and we can use those same modalities in both situations. And we can develop contrast agents or theranostics or diagnostics that do use those modalities, again, preclinically, so we can translate them into the clinic. However, as imaging modalities have developed over the years, there are some modalities that were developed in the preclinical side of things, which are now being developed and actively used in the clinic. And one of those, for example, is photoacoustics. And we'll talk a little bit about that as we go. And then in some cases, there are modalities that are specifically kept into the preclinical world. And that's just because of different ways that have been used to implement those modalities, or they're more invasive in nature, so we wouldn't go ahead and use those in the clinic. So now that we understand preclinical imaging, what are the benefits of preclinical imaging versus in vitro, ex vivo, or in situ studies? So of course, with preclinical imaging, we're using an intact subject. So we're able to study the disease process with the involvement of all the other organs around that disease process. For example, if we're studying a tumor, we can look at the effect of the surrounding organ. If it's an orthotopic tumor, we can take a look at lymph nodes, for example. We can look at the effect of the tumor if there's metastases going into other organs. We have the ability to study the entire organism, and that also includes the immune system. So what's the effect of the immune system on the disease that we're studying? So those can be important things because, again, in the human world, if we have that disease, we have those other organs and immune system being involved in the disease process. Another benefit is being able to study the same animal longitudinally. So of course, most preclinical imaging modalities are non-invasive in nature or would allow us to do a longitudinal study. This allows us to look at the same animal over the course of that study so that we can use it as its own control. This will help to reduce variability, but also helps us to look at the disease mechanism over time. And that can be really important so that we understand from the inception of the disease to the endpoint, what's going on. And if we're using a therapy, how does it affect the disease at the different stages? Also important is the ability to support the 3 Rs, that is to reduce, refine, and replace animals in research. And so specifically here, we're reducing the number of animals needed in a study quite significantly. There may be some refinement and replacement options there as well. And then finally, of course, with imaging, we want to take a look at multimodal imaging. And towards the end of the presentation, we'll talk a bit more about that, what it means for multimodal imaging, and then look at examples of papers where multimodal imaging has been used. So as we move from clinical imaging applications to preclinical, so from mouse to man or man to mouse in this situation, there's a number of considerations that we have to take into account as we're working to design imaging modalities or we're selecting a specific imaging system to use. The first and probably the most important comes down to the size. So what's the size of your imaging subject? What's the size of that imaging target? An example of this would be the human kidney. So let's say we wanted to image the kidneys, kidney disease. The human kidney is about the size of your fist, so maybe 10 to 12 centimeters in size, where the mouse kidney is 10 to 12 millimeters in size. So it's 1/10 the size in the human. So we have to start to look at systems and develop them such that they have an increased spatial resolution so that we can be able to see the structures that are of interest. There's obviously physiological differences between the human and the mouse as well, or other species that we're imaging. One example of that is the heart rate. So again, temporal resolution is going to have to increase because the human heart rate at rest is about 60 to 100 beats per minute, where in the mouse, we're looking anywhere between 300 to 600 beats per minute. So again, the imaging modalities have to have an increased temporal resolution to be able to see the same types of things in vivo that we would see in the human as we would in the mouse. When we're imaging, we're often working to develop novel contrast agents or novel therapeutic compounds. And in developing those, they often are made in small quantities and can be costly. So then we want to make sure that the imaging modality that we're using is quite sensitive to be able to detect those. So we don't have to make large quantities when we are just working to identify a target compound that we then want to track over, track its efficiency and then develop into a clinical agent. And also important when we're talking about imaging any preclinical subject is the animal welfare. So we need to take into account integrated anesthesia if it's required, heating the platform because many animals don't create their own heat while they're under anesthesia, and being able to monitor the physiology of the animal during the imaging session as well. So today during the presentation, I'd like to take some time and overview a few of the different imaging modalities that we represent here at Scintica. I'll talk a little bit about how each one of those modalities works. We'll focus on some of the strengths, weaknesses, or considerations that we have with each. As well, we'll talk about specific application areas and look at some example images. Today, we will focus on MRI, PET, CT, ultrasound, photoacoustics, optical imaging, which would include bioluminescence and fluorescence in this case, DEXA, as well as intravital microscopy. There are, of course, other imaging modalities that we won't talk about today, such as Raman spectroscopy, SPECT, and magnetic particle imaging, as well as many others. So we'll start off with MRI. And as we go through, please know that I'm not going to dive into the physics of these. We have some information on our website that you're welcome to review that dives a lot more into the physics of what's going on. I want to keep this at a very high level, but try to have it so you understand what we're imaging, how we're imaging it, so that you understand a little bit more about the images that we're looking at. So in an MRI system, there's three main components. There's the magnet on the outside, and that's going to create your static magnetic field. The gradients sit inside of that. And these are actually what helps us to localize the MRI signal in three-dimensional space. So this is where we start to differentiate between NMR and MRI. And we actually can create an image using this. And then we have the RF coils, and those sit around the imaging subject. And those are what both transmit and receive signals typically and are interpreted into images. So how does MRI work? In MRI, we're imaging a specific atomic nuclei that has a characteristic. Basically, we want it to be able to act like a little magnet within the MRI system. Most of the time, we're looking at protons. Protons are quite abundant in a living subject, and for the basics of this conversation, let's think of protons as they're attached to oxygen to make water or a carbon chain to make fat. And if you can envision the chemical structure there, you can see that the protons attached to the water are going to be in quite a different environment than when they're attached to a carbon chain where there's lots of other surrounding atomic nuclei to affect the environment that they're in. So outside of the magnet, the protons are going to align in a random orientation. However, once we put the imaging subject into the MRI system inside the static magnetic field, they're all going to align in line with that magnetic field. And then what we're going to do is use the gradients and the RF coils to apply a pulse of energy, an RF pulse, and that will cause the protons to move out of alignment in a very specific way. And then the RF coils are going to receive the signals that we get back as those protons realign within the magnetic field. How they realign and the process that they take depends very specifically on the environment that those protons are located within. So now as we start to look at images, many people talk about different sequences that we take with MR, and many people will talk about a T1 weighted or a T2 weighted image. So let's talk a little bit about what that is. Let's remember that we're looking at protons in a very basic sense attached to oxygen as water or in a carbon chain as fat. And so when we change how we acquire an image, depending on the sequence parameters, which is what a T1 and a T2 weighted image are, we're changing the appearance of the protons. So again, if we think of fat, We can see here the inguinal fat pad, the fat around the testes is bright on a T1 weighted image, where the water is dark. This is the urinary bladder, so we can see that the water is dark. So again, on a T1 weighted image, the fat is bright and the water is dark. On a T2 weighted image, the water is going to be bright, so we've switched. The way that we acquire this T2 image, we speed up the process, which causes the fat to also look bright. So again, if we focus on the images and we also focus on the kidneys, the medulla is dark on a T1 weighted image because water is dark, and bright on a T2 weighted image. How do I remember this? F comes before W in the alphabet and 1 comes before 2. So in a T1 weighted image, fat is bright, water is dark. And on a T2 weighted image, the water is going to be bright. So this comes into play when we start to look at different pathologies and we want to know what we're looking at on the image. And I'll show you some example images on the next slide. So what are some of the key strengths of MRI? There is no limit to the depth of penetration, so we can see all of the different organs and structures within the body. There's relatively high spatial resolution, so we can see the smaller structures on our small imaging subjects. MRI is known to be the gold standard in soft tissues, so we're able to differentiate many of the soft tissues within the body. There's no ionizing radiation needed, as you would see in CT, and we'll talk about later on that we need to consider the effects on our imaging subject over the course of a longitudinal study. The data can be quantified, and of course, it's clinically translatable. When we're talking about different considerations for MRI, there's a relatively low sensitivity to contrast agents, so we need to be aware of that when we're considering what agent to use, for example, gadolinium or manganese-based contrast agent or iron oxide nanoparticles. The systems themselves have traditionally been quite expensive when we talk about superconducting magnets. However, there's an option that we offer here at Scintica that's based on a permanent magnet technology, and that greatly reduces the cost of the system, but more importantly, the siting costs. This system can be installed anywhere in an existing lab without consideration for the surroundings affecting the magnet or the magnet affecting the surroundings. As an example, I can put my computer or my hotel key on top of the system and it wouldn't demagnetize. So you have a lot of flexibility in where you would place the system. One final consideration is that there's relatively low temporal resolution. So moving now onto some images. So here on the left, we can see, again, one of those whole body images. We can identify the liver, the intestines, the urinary bladder, the muscles in the hind limb, and the kidneys as we swipe through. So we can see anatomical and morphological changes. We can image the brain. We can start to see swelling. This is an example of epilepsy. So again, this is a T2 weighted image. So the water is going to be bright. So we're going to see edema. We're going to see swelling as bright on the image. So we can see swelling in the cortex here of this epilepsy model. We can study cancer. Again, we're going to see the tumors often show bright on a T2 weighted image because there's swelling, increased perfusion into those areas. We can study cardiovascular biology, looking at cardiac function as we image different species and look at different cardiac disease models. We can do ex vivo imaging. So here the animal has been sacrificed, we've taken out the tissue, fixed it, and we can invest a lot more time in imaging and therefore get higher resolution images and perhaps use those images to guide the histological sectioning of that sample. Moving now to positron emission tomography or PET. So in the case of PET, we do require the injection of some type of imaging agent. And this would always be, in the case of PET, a radionucleotide. We need something that's going to cause an annihilation event, and I'll explain what that is in a second, to be able to image. We can attach radio-labeled isotopes to small molecules, peptides, nanoparticles, all types of things. And there's lots of different agents being developed currently. And we should always have an imaging agent which is targeted to a specific biomarker of interest or physiological process of interest, something along those lines. So as we have our radioisotope, it's going to decay and emit a positron. As that positron travels through the tissue, it's going to interact with the tissue itself and cause an annihilation event. And in doing so, it's going to emit 2 gamma photons of equal energy moving in exactly the opposite directions. So this is why we have a ring of PET detectors around the imaging subject, so that as those photons emit in the exact opposite directions, the detectors are going to detect what we call a coincidence event. And from there, the system is able to detect where those signals came from and reconstruct that into an image for us. So when we do PET imaging, we need a radioisotope. And when we're doing that and considering what radioisotope we want to use, we need to consider the half-life of that agent and our proximity to a cyclotron. A cyclotron is the device that's typically used to create these radionucleotides. And in so we need to understand how long those isotopes are going to stay active for by considering their half-life. One of the most common agents is FDG, and that's just a radionucleotide and analog to glucose. And so fluorine 18 has a half-life of 110 minutes, and that makes it possible to basically ship the radionucleotide or the imaging agent around a few hundred kilometers so that people are able to use it before it decays too far to not have any activity. But if we're considering something like oxygen, nitrogen, or carbon, those half-lives are significantly shorter, and therefore the proximity to the cyclotron becomes quite important. So again, when we start designing and thinking about our studies, we need to understand if we have access to the radionucleotide in close proximity, or if it's something that we're going to ship in and then create the imaging agent that we're wanting to use. So again, focusing on some of the strengths, there's again no limit of depth of penetration. There's excellent sensitivity. So when we're designing new contrast agents, we only need small quantities. The data is truly quantifiable and of course, clinically translatable. This is very commonly used in the clinic as well. Some considerations is again, as we talked about, having the cyclotron or a generator for the radionucleotides that we need close by. The systems themselves can be relatively expensive, and getting the contrast agent developed or the chemistry involved can also be expensive. We need to think about the ionizing radiation coming from the isotopes, although this is less of a consideration as it would be for something like CT. The spatial resolution, depending on the system, may be limited, and often we need to combine the imaging from PET with another modality such as MRI or CT to provide us with some anatomical context for where that signal is coming from. So here we can see a PET CT image looking at a tumor. So cancer biology, very strong application because we're looking at the increased metabolic activity within the tumor using that glucose analog FDG. We can also look at cardiac function or the lack of perfusion into an area. Again, with the muscle, the myocardium contracting as much as it does, it uses a tremendous amount of glucose. So if there's a myocardial infarction, we'd be able to see an area that perhaps doesn't have as much glucose uptake and identify that. We can look at bone imaging. We can look at the biodistribution of different agents as well as looking in the brain. Moving now to CT. So in CT, we use an X-ray source and we move the detectors around the animal. And this allows us to acquire a full 3D image of the animal. Images can be taken without contrast agent, but there's also CT contrast agents that can be injected to help enhance specific targets of interest as well. So one of the key strengths, again, is no depth of penetration issues, very high spatial resolution, very good temporal resolution, and CT is very strong for imaging the bone and the lungs, and of course, it's clinically translatable. Some of the issues and considerations when talking about CT is its poor sensitivity to those contrast agents if you choose to use them. And primarily, we're looking at bone and lung-filled structures. The resolution of soft tissue is somewhat limited, although some of the new algorithms for processing the images can help us identify some of the soft tissues with a bit more certainty. And of course, we need to start to think about ionizing radiation. Most of the new systems have been developed to limit the amount of radiation that the imaging subject's exposed to. So in most cases, it's not affecting the outcome of your model, and you can image those animals longitudinally, but it is something to think about in your study design. Moving now to ultrasound. So this is a technique that many are familiar with from the clinic. Basically, we have sound waves that are emitted from the transducer, and they reflect from an object, and they're received often by the same transducer. So again, we'll talk about a few other techniques that use sound, but this is sound in and sound out. We can see most of the soft tissues within the body using this technique, but we can also inject contrast agents such as microbubbles to allow us to detect specific biomarkers or to detect perfusion within an organ as well at the capillary level. So when we talk about preclinical imaging, we're always generally talking about high frequency ultrasound. And why is that? When we talk about ultrasound, the frequency of the sound becomes quite important. When we have a high frequency signal, the wavelengths are much shorter, and we're able to resolve two structures quite simply. So higher frequency higher resolution, but they don't penetrate as well. And so we have smaller depth of penetration or lack of depth of penetration. When we move to lower frequency, the wavelengths are longer, they penetrate better, but we don't have as high resolution. So again, high frequency, high resolution, but we need to consider the depth of penetration. So here we can see a carotid artery from a rat. We can see the diagram here, and we can see the image as shown on the screen. Our scale on the right-hand side is in millimeters, so you can imagine how small the structure is in real time. So we need a very high frequency probe to image it. Because it's the carotid artery, we don't need increased depth of penetration. But if we were imaging the heart of the rat, we would need to go to lower frequency to get better penetration. But the rat heart is larger, so we don't need as high resolution. So when we're talking about ultrasound, it's most important to keep in mind your imaging target and how deep it is so that you can select the highest frequency possible that still allows you the penetration to be able to use or to see your target of interest. So the key strengths, ultrasound is typically relatively inexpensive. You can get quantifiable data. There's no ionizing radiation. We can see most of the soft tissues, and there's high temporal resolution. And of course, it's clinically translatable. We need to consider the frequency of the sound, the resolution needed, and the depth of penetration that would be required. Typically, it's used primarily for anatomical information, which we can expand this to molecular information by the use of contrast agents. And we're limited to seeing soft tissues only. So we can't see bone or through bone, and nor can we see air structures like the lungs. So these are some example images. So here we're looking at cardiac function, typical M-mode image, where we're able to image the motion of the heart over time and look at systolic function. We have Doppler capabilities, which allow us to look at blood flow over time. And here we're looking at diastolic function in the heart. In terms of developmental biology, we can image the embryos, confirm pregnancy, watch as they grow. We can confirm that they're viable. Here we're seeing the heartbeat within the embryo. That would indicate that the embryo is viable. If for some reason there was something that was lethal to this embryo, whether it's transgenic or exposure to a specific toxin, we could identify when the animal or the embryo is no longer viable. And we could look at perhaps why. Does the heart not develop properly? Does the brain not develop properly? We can do image-guided injections, so this can help us avoid surgeries. We're able to see here a 30-gauge needle going into the myocardium of the adult mouse heart. We can image tumors, confirm their presence, monitor their growth, response to therapy, and use contrast agents to study perfusion or specific biomarkers. And here we have microbubbles as well, again, to look at biomarkers, look at perfusion, and even do some sonoporation causing gene delivery or drug delivery as well. Moving now to photoacoustics. So in ultrasound, we were talking about sound in and sound out. With photoacoustics, we're talking about light in and sound out. So how does this work? It does require some type of a contrast agent. It can be intrinsic, like hemoglobin, deoxy and oxyhemoglobin respond to different wavelengths of light, or it can be an extrinsic contrast agent, that is injected. So in this case, we're going to use a laser at a very specific wavelength to excite or to have the light absorbed by the specific agent that we're looking to target. When that light's absorbed, there's a very temporary and small rise in temperature, which causes a thermoelastic expansion to take place. This results in the emission of an acoustic wave, which is then detected by an ultrasound transducer. We can then generate images like what you see here. So the strengths of photoacoustic is its superior depth of penetration compared to other optical techniques. It's good temporal resolution, and it is beginning to be more used in the clinic, so clinically translatable. Some of the limitations are you're limited to seeing soft tissue only. So like with ultrasound, we don't see any bone structures or air structures. It does require coupling of the instrument to the subject. And there can be some limit to the amount of anatomical or structural information. So again, you may want to co-register this with other imaging modalities. But as we can see here, we can make up some of the organs like the kidneys, the spleen, and the main vascular structure to be able to identify where we are looking within the different regions of the body. So moving now to optical imaging. In this situation, we're talking specifically about bioluminescence and fluorescence. So they're a little bit different. When we're talking about bioluminescence, we have to have some form of engineered cells that express luciferase, an enzyme that's going to be able to metabolize the substrate that we put in, which is typically luciferin. When those two are able to interact within an oxidated environment, there's light emitted, a chemiluminescent or bioluminescent reaction takes place. In fluorescence, we need to also have a fluorophore of some type. This again can be either intrinsic, something that expresses the fluorophore, like a GFP expressing gene, or we can inject something extrinsic that would be targeted to a specific biomarker that has a fluorophore already on it. And again, here we're going to excite with a specific wavelength of light, and then we're going to look for the emitted light. So a little bit different than photoacoustics where it was light in, sound out. Here we have light in, light out at specific wavelengths. So more recently, there's been some development in both of these. So with bioluminescence, there's been different luciferases and substrates developed to help us progress in the bioluminescent imaging world. And fluorescence, there's a tremendous amount of growth in terms of the number of fluorophores, types of fluorophores that are being used, and the wavelengths that would be used to help combat some of the considerations in using fluorescence in your imaging. So some of the common strengths between the two, they're both relatively inexpensive, relatively high throughput, no ionizing radiation. In bioluminescence, there's excellent sensitivity. Again, both have multiplexing capabilities. Bioluminescence is not translatable into the clinic because we have to manipulate those cells to express the luciferase. However, fluorescence imaging is where we can inject a fluorescent compound to, say, specifically target tumors. We're then able to image that clinically to be able to isolate those cells. And we'll talk about that in the papers that I look at towards the end of the presentation. Some of the limitations, both have limited depth of penetration, relatively poor spatial resolution, but again, we can see structures, so we can see the light coming from the signal with high sensitivity. With bioluminescence, we have to have the substrate and cofactors interacting in the same space, and it's not clinically translatable. For fluorescence, one of the main things to consider here is autofluorescence, and that's where a lot of the work in developing new fluorescent fluorophores comes in terms of moving towards the wavelengths where autofluorescence is not as much of an issue in vivo. These are some example images. Here we have from our Newton system that we sell here at Scintica. So bioluminescence, looking at a lung tumor, fluorescence imaging here, bioluminescence on the brain. So cancer imaging is a main application, neurobiology, biodistribution, as well as cell tracking. The Newton system also has a great 3D capabilities. So here we're seeing a 3D tumor localized in space and the ability to apply an anatomical atlas onto the image to provide us with some context of where that signal is coming from. Moving on to DEXA, or dual energy X-ray absorptiometry. So DEXA works by sending two X-ray beams, one of a low energy and one of a higher energy. And basically these travel through the tissue and we get the X-ray attenuated image. The system then uses an algorithm to be able to differentiate the signal for each pixel. Did it come from bone or did it come from soft tissue? And if it came from soft tissue, we then use the same algorithm to differentiate if it came from fat or lean mass. So the system is able to very quickly, in about 25 seconds for the Insight system that we sell here at Scintica, is able to acquire the images and generate the data that you see here. So we'll get information on bone mineral content, bone mineral density. This is used clinically to look at osteoporosis and preclinically to look at the same thing, as well as other metabolic bone diseases or toxicological effects on the bone, things like that. We can also get measures of body composition. So looking at fat and lean mass, again, looking at changes as disease progressed, looking at metabolic disorders like obesity, diabetes, things like that, or again, in response to a specific compound that you're targeting and ensuring that it doesn't affect either the bones or the body composition when the patient takes it. Here we have some example images coming from a rat. So we can see the X-ray attenuated image with nice resolution, the bone map, as well as the lean and the fat mass. And of course, we can get the quantification that I showed on the previous slide from here. Scan times can be quite fast. Again, on the Insight system that we have here is 25 seconds. Whole body imaging is possible. The ionizing radiation levels are very low, so we don't need to worry about the effect on longitudinal imaging. And of course, it's clinically translatable. Limitations or considerations is that we're looking at 2D images only, and there's low soft tissue contrast, although you can see in this image with the image processing capabilities of the system, we're able to get quite nice definition of the soft tissue. Moving now to intravital microscopy. This is a somewhat new technology to me, and I was amazed by what we can see with it when we started to learn and understand about it and take on the system that we represent called the IVIM system. So here, it's a combination of optical and microscopy. We need to have a window chamber of some type. Here we see a skin fold window chamber, but we have the ability to put those on the skull, on the abdominal cavity, on the thoracic cavity, really anywhere that you want to see a structure of interest. The system can come with either 2-photon or confocal capabilities. And again, we need a fluorophore of some type. This again can be extrinsic or intrinsic. Cells can express a specific fluorescence fluorophore, or we can inject something that we want to specifically image. A laser is used to excite the fluorophore of interest at a specific wavelength, again, confocal or 2-photon. And there's a set of emission filters and microscope objectives that allow us to get microscopic scale images. Here we're looking at the movement of cells through a specific structure, and we're able to track where those cells go through. So here are some amazing, these images always amaze me when I get to see them, of different structures. And we can see here an inflammation response where I believe these are neutrophils homing to a site of inflammation, neuromuscular junction, colon, fat in the liver, with tremendous resolution. So obviously one of the key strengths is the resolution of this imaging modality, the ability to multiplex in terms of being able to see different fluorophores, different structures at the same time. The images are taken in real time, so we're able to see things happen in real time, like these neutrophils migrating or homing to the inflammatory event, and we can make quantitative measures on this. There is relative poor penetration, as with most optical imaging, with 2-photon and confocal imaging, which is where we use that optical window to help us get to the site of interest. A small field of view, we are using a microscope objective, but again, with this system, we can tile different fields of view together to increase our field of view. We need to use multiple lasers for the different excitations. And of course, we need that optical window for some models to be able to see our targets of interest. So now that we've taken some time to take a look at the different imaging modalities, let's start to think about what modality makes sense for you to use in your imaging subject or in your research project. So the primary considerations are going to come into play with feasibility. What's the cost of the equipment to purchase? What are the infrastructure costs for you to install it, the running and maintenance costs, things like that. That's if you're looking at purchasing a new system. What type of information do you need? Do you need anatomical, functional, or molecular? And what type of imaging target, resolution, or depth of penetration is required for you to image the specific target that you're looking for in the specific animal model that you're looking for? And then you need to ask, do I need to image the same animal over time? Do I have the ability to engineer cells to be able to use that specific modality, or have access to or be able to generate the specific contrast agent that would be needed? And do I want to use multimodal imaging, and how would I then move the animal from one imaging system to the next to be able to do that multimodal imaging and co-register those images? So as we move into multimodal imaging or multiplex imaging, what does that mean? So multimodal imaging loosely is defined as 2 or more complementary imaging modalities or contrast reporter agents, which can be used in a single experiment that may be done simultaneously or sequentially. So it doesn't necessarily have to be different imaging modalities like PET and CT. It could be 2 different fluorophores in intravital microscopy, or it could be bioluminescence and fluorescence acquired on the same system. So the goal here is to acquire as much data as possible to track the different biological questions in a more holistic approach. So that would be possible with a single modality. So we want to approach every study that we do to acquire as much data as possible from that animal. But it's also a consideration to start to think about looking at it on multiple scales, going from the whole body to an organ level to a microscopic level. We can see very different things about that disease process at those different levels, and it's important to understand the disease process across the entire spectrum as opposed to just one, for example. So the purpose ultimately is to elucidate the various biological mechanisms of disease so that we can fully understand it. And therefore, as we develop different therapeutic interventions, we can understand the response that the model has to those interventions. If they're effective, what is causing the effect so that we can move forward into the clinic. So these are some examples of multimodal imaging. So more conventionally, something like PET-CT, where we have two imaging modalities combined. Here we have photoacoustics and fluorescence combined. So this is all acquired on the tritome system that we represent here. It combines both photoacoustics and fluorescence. Here we have PET MRI, and we have intravital microscopy, again, using different fluorophores to acquire multiplex image to be able to look at the different structures within the same animal. So finishing up the presentation, we'll take a
Hey everyone, it seems like we've had an internet outage. I will just quickly check and see if we can get everyone back online.
Okay. Hey everyone, so our internet went out of the office, so I apologize for that. I'm just going to share my screen and jump right back in. Okay, bear with me for 1 second while I share my screen again. Always fun when technical issues take over. Okay, so we are on the paper where we were looking at the intramyocardial and the intrapericardial cavity injection of the mice. So here they wanted to measure the effect of the cell injection on cardiac function. So here we have ultrasound images taken with the Prospect T1 that we have at Scintica. And we can see that they used ejection fraction and fractional shortening. And in the intrapericardial cavity injection, they were able to see a more significant improvement in cardiac function than they were in the intramyocardial injection or the controls. They also used fluorescence imaging to be able to show that there was a 10 times better retention of the injected stem cells with the IPC injection compared to the IM injection. They further did immunohistochemistry to find that there was better cell proliferation in the IPC injected animals, less apoptosis, and better vascular regeneration in the myocardium and in the IPC injected animals. Histology was also used to assess the infarct size. So with the IPC animal, there was the smallest infarct size that you see here and the biggest thickening. And in this case, thickening is what we want. The scar or the infarct area, as you can see on the control, typically thins out. So we want to maintain the normal thickness of the myocardium here. And finally, this group wanted to ensure that this was actually translatable into the clinic. So they actually tested the route of injection on a porcine model, and they were able to develop and find a minimally invasive technique that they felt would be clinically translatable into the clinic. So again, in this case, this group used both ultrasound, fluorescence imaging, as well as other techniques like immunohistochemistry and histology to fully understand the disease process, where those cells were going, and what they were doing in this animal model. In the final paper that I'll look at, this is from the Chinese Academy of Science. They wanted to develop a specific multimodal imaging target that could aid surgeons in fluorescence image-guided surgery of glioblastomas. So the challenge here was that glioblastomas are a very difficult tumor. They're often fatal, but in terms of trying to resect the tumor to the best possible outcome, the glioblastoma infiltrates the surrounding tissue as it grows, and it makes resection very difficult. So the surgeon wants to ensure that they resect the entire tumor, but they don't want to resect too much of the tissue as to negatively affect the outcome of the patient. So the goal here was to develop a near-infrared 2 agent, which could be targeted to and specific to glioblastomas. So in this case, they used the folate receptor as a target, which they know is upregulated in glioblastoma. So the folic acid was conjugated to ICG, which is a clinically approved fluorophore, as well as a DOTA chelator so that they could attach copper-64 to do PET imaging. So here we can take a look at the fluorescence images. So here we can see after the injection of the contrast agent, how long did it take to accumulate?
Everyone. Sorry for the up and down. It looks like they're having internet issues at the office. I know they are doing some work in the building somewhere, so I'm not sure if that is causing the issue here. I do apologize for this inconvenience. I will see if I can get a hold of the team on the back end and see if we can get this going as well.
Okay, I think I'm back again. Again, I'm so sorry for the technical problems. Scintica, are you back?
I've been here. I let everybody know that I think we have internet problems at the
Yes,
office. So
we do. We do.
Yeah.
Perfect. Thank you. Okay, so I need to share my screen one more time. And so what we're going to do is jump, I think, straight into the question and answers. So let's move there. Gabrielle, are you back online?
Yes, I'm here.
Perfect.
Perfect. Yeah. So yeah, thanks for the presentation, Tanya. And as you mentioned, we're just going to move there to the Q&A section. And apologies to everyone for the internet issues. It's the first time that it happens so often. But we have the first question, and it is, can you comment on imaging applications in vaccine development, as well as efficacy testing of monoclonal antibodies?
That's a great question, and something that I don't have a tremendous background on. I know with obviously the recent pandemic and everyone working to develop them, there has been a lot of work with different bacterias, different viruses that the viruses and the bacteria themselves have been labeled either with a radioactive nucleotide or a fluorophore, or even some that express luciferase, so that we're able to image the extent of infection, if you'd like, and then in testing the efficacy of different antibodies, be able to inject those antibodies, whether those are labeled as well, to then do multiplex imaging to see if they interact in the same space, or just simply injecting the vaccine and seeing the infection resolve. So those are the situations that I've seen used more recently in terms of using imaging in those specific situations. I, of course, can also think of different ways that you would use, say, ultrasound or MR to look at the effect of the vaccine on the whole body. So toxicological effects, again, of the vaccine once you determine that it's effective. But I think the most common things that I've seen is optical imaging, whether that's using like a traditional optical imaging system like the Newton, or something even like intravital microscopy, where you can get down to the microscopic level and again see the labeled vaccine interacting with the labeled infectious agent, again, whether viral or bacterial.
Great, thanks for the response. The second question is, you mentioned that there was no limit to the depth of penetration of the MRI system. However, when imaging the lungs, we don't see anything. Why is that? They're dark on both the images that you showed, while other structures like the bladder are dark on one and bright on the other.
Sure, no, it's a great question. So if we think back to the MRI, we're looking at protons either attached to water or attached to a carbon chain, such as fat or other structures that make up the body. When we think about the lungs specifically, the alveolar wall is really the main structure of the lungs. And so that's very, very thin. There's not a lot of protons there. So on a normal lung, we don't see a lot of signal because there's no protons there. The air that we're breathing in and out doesn't have protons. However, if you had fibrosis, if you had consolidation of the lungs, fluid buildup, tumors, things like that, we're going to start to see structures filling the lungs. So we can see abnormalities in the lung where the normal alveolar wall doesn't send back enough signal for us to image. So hopefully that answers your question.
Perfect, thank you. The second question is, we have an ultrasound system down the hall. Could I use it to do imaging in the different region of the brain?
It's a great question. So if we think about sound in and sound out, which is how we use ultrasound, that becomes quite challenging to be able to get through the bone with both sound going in and sound going out. The lower the frequency of the sound, the easier we can penetrate the skull, but often we're trying to look for smaller structures. So if you're imaging the brain, a better option would be to consider something like MRI, CT, PET. It really just depends on what you're looking for, but ultrasound is not the best technique. And that's really where thinking about what modalities you have access to, thinking about what you want to image, where it's located, anything that would hinder your ability, and understanding the different imaging modalities comes into play.
Okay, great answer. The next question is, how is photoacoustic per brain review?
Yeah, for sure. So it's a great question. So ultrasound, sound in, sound out. With photoacoustics, we're going to have light in and sound out. So we don't have to worry about the attenuation of the sound wave in both directions. We just have to think about the attenuation of light and then the sound. And so photoacoustics does perform better in the brain. Obviously in the mouse, with the skull being thinner than the rat, we're going to have better success. And we do have examples of that using our photoacoustic system.
Thanks for the answer. The next question is, can I use bioluminescence imaging to image transgenic tumor models?
Great question. So again, if we recall moving back to the bioluminescence imaging, we have to have cells that have been manipulated in some way to express luciferase. So that when we inject the luciferin or other substrate, depending on which luciferase you're using, that they can interact. And so transgenic models, as we know, are spontaneously occurring. We don't cause those cells to be manipulated in any way. So a better way to look at a transgenic tumor model could be with fluorescence imaging with a target that's fluorescently targeted to a biomarker on that tumor, or using something like ultrasound or MR, where you don't need any type of contrast agent to be able to differentiate tumors from the surrounding soft tissue. So again, having that full understanding of what's available to you and how they work becomes super important.
Thanks, Tanya. We have time for one more question, and it's the following. Can you, well, I'm sorry, my apologies. You showed that a window chamber is used for intravital microscopy. I want to study the changes in the inflammatory response over a few days or maybe a week. Could I do that?
Great question. So the window chamber that we saw, they're made of a material that doesn't cause an inflammatory response. And when they're surgically implanted, the animal absolutely can wake up and go back into its cage, wander around and have that disease process or the inflammatory process proceed. And then you can image them again over the course of a longitudinal study. You'd have to check with your animal care people to understand how long it was okay to keep the animal with the window chamber, but those are absolutely longitudinal studies that can take place.
Hey, thank you. So I want to let everyone know that we have reached the end of our session today, and to be respectful of everyone's time, we're going to wrap things up. As mentioned at the start of our webinar, we will be sure to answer any questions in the written transcript, and we will work to get this out over the next few days. I would like to thank Tanya for the great presentation today, and I trust that we have been able to provide you with a better understanding of preclinical small animal imaging modalities and multimodal applications. If in the coming days and weeks you have more questions about the modalities discussed today, I encourage you to contact us here at Scintica, and we will be more than happy to discuss them further. Thanks again to all of you for taking the time out of your day to attend our session, and we look forward to seeing you at a future Scintica event. Have a great day.
Thanks, everyone.